Live. Hello, everybody, and welcome to another session of our Sussex Vision Seminar Series, as always, within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again thank Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from Western Sydney University, Dr. Morven Cameron. Morven completed her PhD on retinal circadian rhythmicity with Professor Robert Lucas at the University of Manchester. And in 2008, she moved to Western Sydney University and Bionic Vision Australia for postdoctoral years, focusing on electrical stimulation of the retinal neurons. In 2012, she was awarded a DECRA Fellowship from the Australian Research Council to investigate gap junction connectivity in the mammalian retina. And following that fellowship, she was appointed to lecturer and nowadays senior lecturer in anatomy and cell biology at Western Sydney University. Her lab is interested in mechanisms that allow retinal circuits to operate across different light levels, from rod and cone circuits to neuromodulator release and the intraretinal signaling and impact of the intrinsically photosensitive ganglion cells, the ipRGCs. In her lab, they employ a plethora of both in vivo and in vitro techniques, electrical recording and molecular imaging tools. And I'm very excited to have Morven here with us, taking us through their findings for her talk entitled Light-Driven Dopamine Release in the Adult and Developing Retina. So without any further ado from my side, please all welcome Dr. Cameron. Morven, the stage is officially all yours.
Thanks so much, George, and thanks so much to the Baden Lab for inviting me. I'm really happy to be here. I will just share my screen. Start here. So it's about 6 p.m. here in Australia. My lights might go off, so hopefully you'll still be able to see me. So today I'm going to talk, as George said, about light-driven dopamine release in both the adult and the developing retina. And this was some work that I started actually during my PhD in Rob Lucas's lab. The project actually preceded me in the lab. And that was a collaboration with Mike Yvonne's lab. And then we published our data. I moved to Australia, did a few other things. And then I picked this project back up in about 2016 when my lab became independent. And I'm going to present some of that data today. So we think this is quite interesting data. Okay, so a quick overview of the retina for our non-retinal biologists. So this is a cross-section of a human eye, and you can see that light enters the eye, it will pass through the cornea and the lens, and this vitreous humor, which is a clear liquid that sits inside our eye. The light will then be focused on the back of the eye onto the retina. So this is a schematic of the retina. So you can see this light coming through the retina, and it actually travels through the majority of the circuitry of the retina before it hits these photoreceptors, what we call classical photoreceptors at the back of the eye here, rods and cones. And so rods and cones have slightly different functions. So rods are very sensitive to light and govern our vision under really dim light conditions. Cones are less sensitive and so govern our vision under brighter light intensities and give us the rich experience of the world, such as high visual acuity and color vision. So these photoreceptors will communicate, they will detect the light and then communicate that light information through the circuitry of the retina to ultimately end up in the retinal ganglion cells, which will project their axons down the optic nerve to the brain. So the retina is actually part of the brain. It's not just central nervous system, it is an extension of the brain. It was quite surprising just over 20 years ago that it was discovered that these were not the only photoreceptors in the retina, and that another photoreceptor was discovered as a subset of these retinal ganglion cells. So these are the intrinsically photoresponsive retinal ganglion cells. And so if you completely remove the rods and cones, you still have light responses in these cells. And so while rod and cone photoreceptors are photoresponsive, they can respond to light via rhodopsin and conopsin respectively, ipRGCs, as we call them, express the opsin melanopsin. So sometimes these are called melanopsin-containing ganglion cells or just melanopsin cells. Okay, so my title says I'm going to talk about dopamine. So I think we've heard a lot about dopamine in the pop science social media recently, and you'll hear that it's to do with motivation and reward. But of course, it's really just a neuromodulator, and so it modulates neural circuits. The retina doesn't have anything to do with reward or motivation, its role in the retina is going to be to do with detecting light, which is the retina's main role. So one of the most important things for this talk to understand is that dopamine is released in response to light. So dopamine levels are low in the dark and high in the light. And dopamine does a few things in the eye and the retina, but one of its best described and probably its predominant role in the adult retina is that of light adaptation. So we can think about light adaptation in a few different ways, and I think perceptually, the one that most people have experience with is desensitization. So desensitization is the experience where you've been in a really bright light environment. So you might be at the beach in Sydney, for example, and then you try and go into a dim light environment. So, a dimly lit bar, for example, and you really can't see anything at all. So you feel like you've lost something from your vision, and you've lost your dim vision. But we don't think about the other side of it, where our vision while we were on the beach, our retinas have really optimized our perception of increments of light over a background. So what do I mean by this? And I don't think it's something that is immediately obvious perceptually to people, but we can track it really nicely using a technique called the electroretinogram. So the electroretinogram is a commonly used clinical technique. This is an example in humans. We work on mice, but we can do this in both species, where you record the electrical potential from the cornea via a non-invasive electrode, and you can record the electrical response of the retina. So we can track the light adaptation in our mice really nicely and show this visually using the electroretinogram. So what we do is we keep these animals in the dark for a long time. So they're very, very dark adapted, about 18 hours. And then we switch on a really bright background light. And on top of this background light, we superimpose an even brighter flash. And so the retina must tell the difference between this background light and the flash. And so what you see when you first turn on this background and give the flash is that the response is quite small to that flash. And the longer that the background light is on for, this response grows. So this is what I mean by optimization of increments of light over background. So your vision is getting better to determine the difference between these two quite bright lights through the process of light adaptation. And it's thought that the release of dopamine is one of the primary signals that allows this light adaptation over this 15 to 20 minutes to occur. Okay, so where does this dopamine come from in the retina? So dopaminergic cells are amacrine cells, and so they sit in the middle layer of the retina, so in the inner nuclear layer. So this is a dopaminergic cell in a mouse. This is a cross section of the retina, similar to this. And you can see that we've stained it here with one of the enzymes for dopamine production. These are thought to be the only cells in the retina that produce dopamine. And you can see that they have this really extensive plexus in what we call the off sublamina, which is this layer here. And then when we look down from this direction on these cells, you can see that they have this really incredibly dense plexus of dendrites surrounding the neighboring cells. So you can see the holes here are the neighboring cells. So these this absolute net of dendrites completely encircles all of the neighboring cells, allowing these cells to really have a lot of influence on their neighboring cells. But dopamine is also released by volume transmission, so it can diffuse throughout all the layers of the retina and also into the vitreous, which we'll talk about soon. And every single type of neuron in the retina has been shown to express dopamine receptors. So it can have this really global effect on the circuitry of the retina to allow it to adapt the retina. So these dopaminergic cells form this plexus across the entire retina. So there really isn't a part of the retina that doesn't have this plexus in it. But the cell bodies are really quite sparse. So we only have about 40 cells per millimeter squared in the mouse. That's only 600 total cells in a whole mouse retina. But this plexus really covers the entire retina. Dopamine is released in response to light, and we can show that all of these dopaminergic cells are activated. They're showing a marker of neuronal activation here, which is C-fos. In response to light, we get 100% activation of our cells. So they're really strongly driven by light. So I think the retina really presents a good opportunity to really define the mechanisms of dopamine release, because we have this circuit here where we can apply a really defined light stimulus. We can really apply any sort of light stimulus we want. And then we have a neuronal circuitry that detects this light, and then we'll pass that light information on to our dopaminergic cells, and then they will release dopamine. So using this circuit, we can ask a few questions, such as which photoreceptor is driving this light input into our dopaminergic cells. And so we have three options there, so rods, cones, or ipRGCs. We can also ask, how does the photoreceptor convey this light information to dopaminergic amacrine cells? So this is a really important question in the retinal field. And of course, over the last 100 years, we know a lot about the retinal circuitry, although I think we're still finding more. And we can also ask, how is that dopamine released once the signal reaches these cells? And I think while these two questions are more retinal biology specific, I think the question on how dopamine released is something that's more broadly interesting to the rest of the CNS, because dopaminergic amacrine cells are thought to function in a really similar way to other dopaminergic cells in the rest of the brain. So I'm going to talk today about we have quite a good handle on which photoreceptor is driving this response, and we know some of the answers to these questions, but I think there is a lot more work to do here, and I think this nicely defined circuit gives us the opportunity to do that work. Okay, so if light information is getting to these dopaminergic cells, and they are not themselves intrinsically photoresponsive, how are they getting this light information? So there's a few ways that we can do this. And this was an approach that was taken by Doug McMahon's lab initially, and then Dao-Chi Tsang's lab laterally, where they can record from these dopaminergic cells. They can record the electrical potentials from these dopaminergic cells and shine light. And they have shown, using a variety of transgenic animals, that light inputs can be recorded in these cells that originate in rods, in cones, and also in ipRGCs. So it seems here that all three photoreceptors are contributing light information to these dopaminergic amacrines. But what we were interested in, and this is becoming a bigger question, I think, certainly in retinal biology, but also in other areas of neuroscience, is does this electrical response that we can readily measure in the dopaminergic amacrine cells, does that actually equal dopamine release? And so I think we can talk a little bit about this at the end, but I don't think this is necessarily true, this textbook idea that spiking will always equal neurotransmitter release. Okay, so how do we actually measure dopamine release? So we do a few different methods. We do in vivo methods. So these are mice that, again, similar to our ERG protocol, we keep them in the dark for a long time. So they go in the dark at dusk, and they're in the dark overnight. And this is complete darkness overnight. And then we keep them in the dark until about subjective midday the next day. So it's about 18 hours in the dark. And we either keep them in the dark, or we'll give them a 1 hour light pulse of approximately 6 log photoisomerizations per rod per second. So if this is not a unit that you're familiar with, most people aren't, this is really bright light. This would equate to perhaps not full sunlight, but maybe a slightly overcast day in Sydney in autumn, like we have today. So it's pretty bright light for a mouse. We also do not apply any mydriatics to dilate the pupil, and we don't give any anesthetics to the animals. So the animals are free moving, and that gives us a little bit more variability in the amount of light that they get. They're in a light pulsing box, but they can close their eyes. They can hide from the light a little bit. So they will get a little bit of variety in the light. So we light pulse these animals or keep them in the dark, and then we'll remove either the retina or the whole eye, and look at the dopac to dopamine ratio. So this is a really commonly used ratio in the field, and it reports the turnover of dopamine in the retina. I won't go into great detail about how it works, but an increase in this ratio shows that you've got an increase in dopamine release. But we also started recently doing a new in vivo measure where we look at dopamine release into the vitreous. So you'll see a slight difference between this schematic and the schematic I showed of the human retina. So mice have this enormous lens and very little vitreal fluid. But the idea here is that any dopamine released in the retina, so the retina's in the red here, will diffuse out into the vitreous, and then we can remove that vitreous. We get about 2 microliters of vitreous, and then we can measure dopamine and dopac via ultra high performance liquid chromatography with tandem mass spectrometry. So I'm not going to talk about that at all today, but that's how we measure it. So these methods work really well. So this I'm showing here is dopac to dopamine ratio in the retina. So you can see in our dark adapted animals, we have a sort of basal level of dopamine turnover, and then a substantial increase in response to light. Similarly, we get the same response when we just look at whole eye. And then when we look at vitreous, we get a much more robust increase. So almost a 10-fold increase in the concentration of dopamine in the vitreous compared in the light compared to the dark. So I'm going to use these measures somewhat interchangeably in this talk. I've always labeled which ones I'm talking about at any given time. They broadly give us the same result. However, there are slight differences between them, and we think that dopamine concentration in the vitreous is really the best measure that we've tried so far. Okay, so these are all in vivo measures. So how do you know that this dopamine is actually coming from these dopaminergic cells? I showed you some beautiful pictures of dopaminergic cells, but the eye is still attached to the body, so it might not necessarily be coming from these dopaminergic cells in the retina, but we can look at that by removing the eye from the animal, cutting off the cornea and the lens, and then submerging the retina within the eye cup in artificial cerebrospinal fluid. And then we can light pulse the retina and record and then measure the amount of dopamine that is released from this retina into this artificial cerebrospinal fluid. And so this again works really nicely. So you can see that we have this robust increase in dopamine release in our light pulsed conditions. So having this in vitro method is also really useful because it allows us to apply pharmacology to the retina to block known pathways. So the first drug that we used was LEP4. So LEP4 blocks the communication. It's an mGluR agonist, and it will block communication from rods and cones to their respective on bipolar cells. And so when we apply this to our eye cup preparation, you can see that light-induced dopamine release is completely abolished. So this was our first indication that maybe it's rods and cones that are specifically driving dopamine release, because as soon as you block the rod and cone input, you lose the light-induced dopamine release. And this hypothesis was strengthened by the fact that this ipRGC input to the dopaminergic amacrine cells that has been previously described, it persists in the presence of LEP4. So we're not getting dopamine release despite still having this ipRGC input. So we wanted to look at this in more detail using specific knockouts of photoreceptors. So you can see we've got our wild type light-induced dopamine release here. And the first animal that we looked at was the rodless, coneless animals. So they lack all rods and all cones. However, their ipRGCs are still really functional in these animals. But what we saw was that light-induced dopamine release is completely abolished in these animals, indicating that ipRGCs are no longer, are not sufficient to drive this light-induced dopamine release. Similarly, if we knock out melanopsin, so we leave rods and cones where they are, and we knock out melanopsin, so now our ipRGCs are no longer intrinsically photoresponsive, you can see that we get totally normal light-induced dopamine release, and they don't look any different from the wild types. Okay, so the rodless coneless animals are a degenerate animal, a degenerate retina. So it could be that there's a lot of rearrangement or degeneration in the retina. So this might not be the best model to look at. Similarly, the melanopsin knockouts have been shown to have developmental issues that might change circuits. So we wanted to look at this in a bit more intact retina. And so with the help of Nina Milosavljevic from, at the time, Rob Lucas's lab, we did some chemogenetics, so using GQ dreads. So chemogenetics is a way of activating specific subsets of neurons with a drug. And this is really useful for retinal biologists because it allows us to activate subsets of cells in the retina without using light. And so we injected Flox GQ dreads unilaterally into melanopsin Cree heterozygous animals, and we can see expression of these dreads receptors in ipRGCs. And what's nice about the chemogenetics is that we are able to systemically apply a designer drug. We use CNO, and that designer drug will specifically activate just the cells expressing this dreads receptor. And so you can see when we inject that, we get this C-fos, which is the marker of neuronal activation. We get C-fos activation in these dreads expressing cells. Really handily, because the ipRGCs drive the pupil light response, we are able to get a significant pupil constriction in response to this drug, which activates these cells all in the dark. So we have a nice method here of selectively activating ipRGCs. We can tell that, the majority of the ipRGCs in these retinas of just one eye of the mouse are activated, and we're not activating rods and cones. So does this elicit dopamine release? So you can see here that our melanopsin Cree animals, which we use for the DREAD study, have completely normal light-induced dopamine release. But when we activate DREADs unilaterally, you can see that the DREADs activated, the concentration of the dopamine in the DREADs activated eye does not differ to the concentration of the non-activated eye, and these are the same as the uninjected eyes in the animals in the dark. So I feel quite confident now with all of these measures saying that ipRGCs are really not driving dopamine release. However, as we showed you before, they have this significant input into dopaminergic amacrine cells that can be measured. And so how do they have an input and not release dopamine? So I'll talk about that a little bit later in the talk. But at this point, we can see that rods and cones are driving dopamine release, because if we lose them, we're losing our light-induced dopamine release. But is it both, or is it just rods or just cones? So we looked at that in another model. So the GNAT1 knockout animal, which is a rod inactivated animal. So they lose the majority of their rod function. And you can see that there's a significant decrease in our light-induced dopamine release. However, a small amount of increase in our dopac to dopamine ratio, this is the retina, is still observed in these animals. So it's possible that cones are driving a small amount of this light information to these dopaminergic amacrine cells, but it seems like rods are really predominantly driving it. A small caveat to this result is that the GNAT animals actually still retain a small rod response. So we actually published this some time ago now. So it's possible that this remaining light-induced dopamine release is actually being driven by the remaining rod response in these animals. Certainly, when we inactivate cones in the GNAT2 animal, we don't see any difference between the cone inactivated animals and the wild-type animals. Further, if we inactivate cones and remove melanopsin, so these are essentially rod-only animals, we still get completely normal light-induced dopac to dopamine ratio. We come to the conclusion from these transgenic animal studies that ipRGCs are neither necessary nor sufficient to drive dopamine release. Rods can drive dopamine release without cones and without melanopsin. And cones may fine-tune the amplitude of this dopamine release, but I think we have to do more work to look at their contribution. But it's really predominantly rods that are driving it. Okay, so if it's predominantly rods that are driving this response, we would expect the release of dopamine in response to light to match the sensitivity of rods and also to match the sort of temporal properties that we understand rods to have. So the answer to this question, the answer to both of these questions is no, it does not match that of rods. And so what we found when we did the Irradiance response for dopamine release, and you can see increasing light intensity on the x-axis here, is that we don't get significant light-induced dopamine release until really quite bright light intensities, so above the cone threshold. So we've got about 5 log units where rods are signaling and not causing significant dopamine release. So we were pretty surprised by this, and also by the fact that when you apply dim light, that you actually get a suppression in the amount of dopamine that's released. This is also the response of, this is also the response to an hour of light pulse. And we know that rods really saturate under bright light conditions. So we wanted to know what the temporal release of dopamine looked like. And we've done this very recently with our vitriol measurements. But of course, as I told you, our dopaminergic cells sit in the middle of the retina. And to measure the dopamine in the vitreous, we have to wait until that dopamine has diffused out and got into the vitreous. And so we didn't really expect to see it very quickly in the vitreous, but again, we were wrong about that. And you can see that we get significant we get a significant increase in the concentration of this dopamine in the vitreous after just 1 minute of light stimulation. It takes us probably 30 to 40 seconds to get the vitreous out, so it's maybe 1.5 minutes, but that's a really short amount of time for the dopamine to be released and diffusing into the vitreous. So it happens really fast. It then plateaus about 15 to 30 minutes, and it just stays up until you switch the light off. And then when we switch the light off, we switch it off an hour here, you can see it comes down pretty quickly. So it's really an on-off signal where you switch a light on and it goes up quickly, it stays up, and then you switch it off and it goes off again. And that's not how we think about rods functioning either. We think that they saturate really quickly, and then bright light signals can be recovered in rods, but it takes some time, and the response does not look like this. We also extended this out to about 6 hours, and we don't see any difference between the concentration that we see at 6 hours versus 10 minutes. So this is a really sustained response. Okay, so I think the main questions here are, how do rods drive such a high threshold and sustained response? And then also, as I talked about earlier, why does this input from ipRGCs not cause dopamine release? And might these two questions be related? So I'm going to go over in the next few slides some of the hypotheses that we have formed to try and explain this, one of which I will show that we got very wrong. So we took inspiration from this great paper from Will Grimes from Jeff Diamond's lab, where in another type of amacrine cell, so the A17 amacrine cell, you get these microcircuits at these varicosities on the dendrites of these cells. So what they showed was that you can have a synaptic input at this varicosity, for example, and that synaptic input will cause a calcium response. But if you measure the varicosity, the neighboring varicosity on the same cell, just 30 microns away, it has no calcium response. And so this led them to believe that you have these microcircuits, that you have a synaptic input and you can have synaptic release very locally just at one varicosity. And that within this one A17 cell, that you could have over 100 independent microcircuits functioning in this cell. And similarly, this system has been reported in dopaminergic cells, not retinal dopaminergic cells, but those in the striatum. So they've shown that, of course, the distances are a bit different in the brain. So the somas sit in the midbrain and the terminals sit in the striatum. And so what's been shown is that you can have really local release from these varicosities on the dopaminergic cells, where you have a synaptic input from acetylcholine cells that cause dopamine release just at this one varicosity. And so we have these varicosities on the dopaminergic cells. This is an example of some really large ones. They also have some smaller ones too. And we thought, well, maybe these dopaminergic cells are working in a similar way. So our hypothesis that we published in our paper here was that at these varicosities, maybe in the dark, we've got no inputs, so we have no rod activation at these complete darkness. And so you have a sort of basal firing rate and some basal release of dopamine. However, when you turn on a dim background light, you have a rod input that causes suppression of this of these dopaminergic varicosities. It could possibly cause some excitation too, but certainly the inhibition must outweigh that excitation. And then you get actually a reduction in dopamine release. And then at some point, this excitation may tip the balance and overcome this inhibition to cause this high threshold and high amount of dopamine release that we see at bright light intensities. So to look at this in more detail, we turn back to our in vitro model and to look at this inhibitory input. And we know that dopaminergic cells get GABAergic inputs. So we tried blocking with the GABA-AC blocker picrotoxin. And what we can see is that under these conditions, we see a significant increase in dopamine release. So relieving inhibition from these cells is enough to cause this large amount of dopamine release. So we might be onto something with this inhibitory input idea. We also wondered about spiking. So this spike here, you can see is this necessary for dopamine release? Can it drive dopamine release on its own, or do we need synaptic inputs? And so we looked at that using TTX. And what you can see is that when we block with TTX, which will block sodium channels, you can see that light-induced dopamine release is abolished in these under this TTX block. And then conversely, if we increase spiking, and we can do this by applying 15 millimolar potassium chloride, we don't see an increase in response to increased spiking in dark conditions. So increased spiking does not increase the amount of dopamine release. However, when we pair that increase in spiking with light activation, we see a significant increase above that of the light conditions. So what this suggests, and it's certainly not categorical, but it does suggest that spiking is necessary for dopamine release, but not necessarily sufficient to drive dopamine release. So that would fit with our hypothesis quite nicely. The other interesting pharmacological result that we got was that when we block AMPA kainate receptors with CNQX, that again abolishes light-induced dopamine release. I think that was quite accepted if we think about the circuitry of the retina. However, what we were surprised to see was that if we block NMDA receptors with AP5, while we don't completely abolish light-induced dopamine release, we significantly reduce it. So this led to my great idea about this hypothesis that perhaps we're seeing coincidence detection. And this, of course, could be at the level of the dopaminergic varicosities, but maybe we need two inputs that coincide to cause this dopamine release. And so, you know, you could have an input from rods and an input from ipRGCs, and that you need both to have dopamine release. Okay, this doesn't quite explain our result that rods can drive dopamine release on their own. However, we know that rod information can be conveyed through ipRGCs. Sorry, I forgot to say, ipRGCs certainly do. It's thought that their input into dopaminergic cells is driven by NMDA receptors. But yet the rod photoreceptors, we know that they convey light information through ipRGCs. So it's possible that this coincidence detection is going on even when we are lacking melanopsin, but the ipRGCs are still there and intact. So I thought this is a nice hypothesis. People in the field seem to like it, but there's a nice way that we can test this hypothesis, and that's by removing these ipRGCs, and that would then remove this arm of our putatory coincidence detection circuit. Okay, and so I was at NIH last year for a couple of weeks, and I managed to talk a couple postdocs into helping me with this. So Corinne Bayer from the Hattar lab and Fran Nadal-Nicolas from Wei Li's lab, I convinced them to take some vitreous, to light pulse and take some vitreous out of the OPN4 DTA homozygous animals. So Samer has quite a few different DTA animals, OPN4 DTA animals. These ones completely kill M1 to M3 subtypes of ipRGCs. It's possible that some of the other subtypes still remain in these animals, but it's thought that the M1 subtype is the one that inputs into dopaminergic amacrine cells. So these cells should be completely lost in these animals. So Fran and Corinne light pulsed these animals. They did some wild type controls as well, and then they posted the vitreous over to Australia and we measured them. And what we found was that in their wild type controls, we saw pretty much the same concentration as we see in our Australian mice. So that gives us good confidence that this is a really reliable measure of dopamine release. And so what did we see in these animals that are completely lacking ipRGCs? Well, we saw completely normal light-induced dopamine release in these animals. So even I was pretty surprised about this, that you can completely abolish ipRGCs and light-induced dopamine release is completely intact. And actually, it looks like there's even more dopamine release in these animals than there are in the wild types. And there's quite a simple explanation for that, because of course they lack their ipRGCs. I told you we don't use any mydriatics to dilate the pupils. So our wild type animals, their pupils will look like this, whereas the DTA animals, their pupils will be, almost completely dilated. So yeah, they have completely normal light-induced dopamine release. And my hypothesis about this coincidence detection was not correct. Obviously, it could be occurring. It's just not occurring through ipRGCs. Okay, so I feel like I'm hammering home the point now, but ipRGCs are not driving dopamine release. And I will put the caveat here that, we can't exclude the fact that ipRGCs might be driving a lower level of synaptic release that we just can't measure. But this high amplitude increase in dopamine in response to light that we see in the wild type animals is driven by rods and not ipRGCs. So I think coming back to this hypothesis, this is the most likely explanation of how the rods are doing this, that they have both an inhibitory and an excitatory input to these cells. And it's really the balance between these two inputs that explains how rods are able to drive this response that really doesn't look like a rod response. And why spiking from ipRGC that is causing spiking at the soma of these dopaminergic cells, so input from the ipRGCs, why that might not be necessarily driving dopamine release. And this was actually a hypothesis put forward by David Marshak in a chapter he wrote in 2001, before it was really accepted that ipRGCs were a photoreceptor in the retina. So yeah, I think this is the most likely hypothesis so far. Okay, in the last 10 minutes, I'm just going to talk about some work we're doing in the developing retina. And it's along the same lines, but the developing retina is really interesting. So in our adult retina, we have our rods, cones, and ipRGCs. I don't have them labeled here. Sorry, in the adult retina, we have rods, cones, and ipRGCs, whereas in the developing retina, certainly in mice, they don't open their eyes until postnatal day 13. And we don't think rods and cones are functional until at least after postnatal day 10. But ipRGCs are actually active before birth. So even when the mouse is in its mother's uterus, these ipRGCs are picking up light information and communicating that to the retina and the rest of the eye and the brain. So we wanted to know, they don't have rods and cones. So are melanopsin cells driving dopamine release at this point? Do we even have light-induced dopamine release in these animals? They probably don't need to light adapt at this age if their rods and cones are not functional. So I showed you earlier in the talk that this marker of neuronal activation, C-fos, when we light pulse the retina, we can activate 100% of these dopaminergic amacrine cells in the adult. And so we used this method to look at our developing animals. So at postnatal day 8, and this is the same paradigm as I showed you before. So it's actually a 90-minute light pulse. So that's the peak for the C-fos activation. So we light pulse these animals at P8. And what you can see is that we get C-fos activation in the retina. You can see all these here. And this is driven by melanopsin phototransduction. But what you can also see is that none of this C-fos is expressed within these dopaminergic amacrine cells. So absolutely no co-expression at P8. At P12, so this is just one day before the mice open their eyes, you can see that we have still lots of mainly melanopsin-driven C-fos activation, but the majority of our dopaminergic cells still do not co-localize to co-express C-fos. However, we do start to see a few. So you can see a few in this retina here. We see 10, and they're all in the dorsal retina. So we are getting activation just this one day before eye opening. And then if we look at P14, which is just one day after eye opening, we get 100% activation of these dopaminergic amacrine cells. So, you know, I'm not a developmental biologist, and maybe this sort of thing happens all the time, but I find it absolutely fascinating that you have this switch where that light information just suddenly is allowed to be communicated to these cells at P14. And just 2 days before, pretty much every cell in the retina is not able to receive this light information. But it also argues that, you know, we've got significant melanopsin phototransduction prior to eye opening, but it's not communicating that information to the dopaminergic cells. So we think the situation is the same in development. The melanopsin cells are still not important for dopamine release. So speaking of dopamine release, what does dopamine release look like in these animals? And so I think the first thing to notice is that in our young animals, we have a lot more dopamine in the vitreous than we do in our almost adult retinas. So you can see that it declines as the animal gets older. So you can see at P30 when the retina is thought to be pretty much in its adult state, you've got much less dopamine than you do at P8. So this makes us think that dopamine is probably pretty important for development at these early ages. When we light pulse these animals, you can see that perhaps not surprisingly, we don't see any difference when we give a light pulse at P8. But at P14, where I showed you that 100% of our dopaminergic amacrine cells are activated by light, we still don't see an increase in the dopamine, in the light-induced dopamine release. At P16 and P19, we see a trend for an increase in dopamine release in response to light, but it's actually not until P30 that we see this significant increase in response to light. We also measure a metabolite of dopamine, which can sometimes be a bit of a more sensitive measure of dopamine release. And so we did that in the vitreous of these animals too. And we saw a lot less variability with this. And then we were able to see a very small increase in DOPAC concentration at P16, which gets bigger at P19, and then even bigger at P30. So it seems like this light-induced dopamine release really develops following eye opening, even though the cells are really activated completely immediately after eye opening. And if we compare this to the complexity of this really dense plexus that I talked about in the introduction, it really correlates with this increase in light-induced dopamine or DOPAC production. So you can see here, we don't have a very dense plexus at P14. It's starting to develop. It's getting a bit more dense at P16. It's getting a lot more dense at P19. And then by P30, you have this really dense plexus. And I think this really argues that the light-induced aspect of dopamine release is really dependent on this plexus. And I think this argues a bit more for the idea that these microcircuits on the varicosities, which would be on this dense plexus, are driving this dopamine release. Okay, I think I'm right on time. So to summarize, ipRGCs are not driving dopamine release in either adult or developing retina. Dopamine release seems to be defined completely by rod input, and that rod input is really high threshold, but I think we've got lots more work to do on the really dim light intensities, and we're planning on doing a much more comprehensive irradiance response to look at these dim light intensities in more detail and the circuits that might be driving this suppression as well. Cones may make a contribution to the amplitude of the response. I think we've still got lots of work to do here. And then the electrophysiological response of the soma is not correlated with dopamine release. And I think this is becoming a bigger theme in retinal biology, that what you record at the soma really doesn't tell you the whole story, and that there's a lot going on elsewhere in the cell that we can't appreciate by just measuring from the soma. So I'd like to acknowledge the people who did a lot of this work. So the majority of what I talked about was done by my former PhD student, Victor Perez Fernandez. The development work that I talked about at the end was done by Vrinda Jain, who is a MD student in the lab. She really didn't spend a lot of time in the lab, but got a lot done. Philip Liang is also an MD student. He's done a huge amount of work on optimizing the methods that we're using to measure dopamine release, and certainly being able to measure in the vitreous is a game changer in terms of like the sensitivity that we have, and it's much more sensitive measure than the ratio that we were using before. Sush, my former PhD student who has just submitted her thesis, she did the work on the dreads, and then the people from our mass spec facilities, so the chemists, I think chemists and biologists should really collaborate more. It's been very fruitful. When I started this project, I just was like, can you just measure the dopamine? And I don't want to know anything about it, just go and measure. But I had to learn more about it, and it has been really fruitful. And my collaborators at NIH, and some funding from the Ainsworth Medical Research Fund, and the two MD students had some summer scholarship funding too. Thank you very much. I think you're on mute, George.
Yeah, now you can hear me, I hope, right? Thank you so much, Morven, for this fantastic presentation of your very meticulous efforts to tackle the neuromodulator release. I can't help but be a little bit jealous that you have, all these very precise approaches, like from genetic lines to chemogenetic perturbations to pharmacology to all these fancy mass spec tools. I was so impressed with the resolution that you have with these vitreous measurements, both like the resolution itself, but also the kinetics of the release. And my first question was like, it appears that it very fast goes down as well after you switch off the lights, right? So do you know anything about the uptake or the washout of dopamine?
We don't. I mean, there's certainly dopamine transporter in the retina. We've done a little bit of work on that. We have some collaborators that give their mice cocaine, and cocaine's a really potent blocker of the dopamine transporter. So certainly that can affect dopamine levels. It can affect dopamine production as well. But no, it's really surprising to me that it goes down so quickly. Right. Because when we take the vitreous out, occasionally it takes longer than the 30 seconds that it takes us to take it out. But usually the concentration doesn't change much. And we don't think there's enzymes in the vitreous that are breaking it down. So yeah, it must, I don't know, it must diffuse back in the retina and be broken down. But yeah, it really declines very quickly.
Right. Thank you very much for addressing this. Before I move on to the questions that start appearing in the chat, I would like to remind our audience that at some point we will be going offline. So I have already gone ahead and posted the Zoom room link there, and people are already joining us. So in case you want to continue in the conversation, please make sure you save this link, because when I stop broadcasting, this link will disappear temporarily. So the first question in the chat is from Leon, and I think it's a great question. I also want to ask something similar, so I'm biased in characterizing it. So Leon asks, do you think that cones might play a greater role in controlling dopamine release in diurnal animals?
Yes, that's a good question, which Wei Li and I are looking at the moment in the ground squirrel. Yeah. So yes, possibly. I won't tell you our results yet because they're quite preliminary. But if you think about dopamine as separating the rod and cone pathways, with its effects on gap junctions, it really removes the rod input from the cone pathway. Rods are always using cone pathways. So that, co-optedness of the rods use the cone pathways, the dopamine's removing that. And so if you're a diurnal mammal and you're not really using your rod pathway anyway, do you need dopamine to be released in response to light to separate those two pathways? So I don't know. I don't know if it would be very important in diurnal animals.
Right, but I think it's really important that you are looking into the cone-dominated ground squirrel for that, because it's also on a very similar evolutionary scale. A question that I have on similar grounds is like, do you try to do these experiments at different times of the day, or are they always in the morning?
Yeah, so we've done midday and midnight, and yeah, we get some kind of weird results with that. We don't have any way of reversing our animals at the moment, so it's quite difficult to do these experiments because you have to come in at 2 a.m., which is our mouse's midnight. We don't see a difference at the moment. Our mice are black 6 background, so they don't have any melatonin. Melatonin is supposed to be very important to entrain the circadian rhythm of dopamine release. So dopamine release is supposed to be when you have melatonin, there's more dopamine release in the day and less in the night. We don't see that in our animals, which is not surprising because they don't have melatonin. So we're really interested to use our techniques to look in animals that have melatonin. In terms of dopamine release, we don't see any difference either. So yeah, not yet.
The next question appearing in the chat is from Subha Sandra Yadav, and I'm sorry if I'm mispronouncing the name. What is the pathway for rod-driven dopamine release? Rods to rod bipolar cells, to A2 amacrines, to type 6 bipolars, to type 6 bipolar cells at the off layer, to then the dopamine amacrine cells? It sounds strange given the paucity of ectopic synapses on bipolar cells.
Yes, but I think that's probably the pathway, although, you know, you have to have this inhibitory pathway too. So certainly like the inputs to dopaminergic amacrine cells have been pretty well characterized from type 6 bipolar cells into the dopaminergic amacrine cells in the off layer from the en passant synapses. And that I heard Dave Berson's talk, you know, they think of that as kind of the luminance coding accessory on pathway, which I think is a really cool idea. So yeah, we think that's probably the pathway. There's some indication that the input, I know in the primate retina, some of the work that David Marshak did, they said that the cone bipolars that input the rod information do not have inputs from A2s. And so their idea is that it's rods to cones to cone bipolars and then into the dopaminergic amacrine cells. But then why aren't cones more important?
Right.
I think that's another question too. But yeah, that's the pathway that we think it's traveling through.
Great. And as we are approaching the 1 hour mark, I will post the Zoom room link again in case people want to join us. And this will be the last question from my end, as there are no more questions appearing in the chat. So the question I have is like, with respect to the latest part of your talk about the development, it appears that there is a trend in decreasing the dopamine levels at dark with development, right? So would this add evidence to the inhibition hypothesis, or is it completely unrelated?
Yes. I hadn't thought about that much. Yeah, no, that's a good point, that you would have more inhibition as you went on. We haven't really looked at the inhibition under completely dark conditions yet. So the picotoxin stuff I showed was under dim light. So it would be quite interesting to look under dark conditions to see if that inhibition is still important without any rod activation. So yeah. I think you could be right.
Great. And as people are already here, I would like to thank you once again, Morven, for this fantastic talk. And let's continue offline. Thank you very much.
It's great to be here.